Hello everyone, welcome back to your channel Bioexam and Research Support by Archana. I'm super excited to start this new series on recombinant DNA technology for CSIR NET Unit 13 today. And we are jumping into a core part of genetic engineering restriction endonucleases. These enzymes are like scissors for DNA and trust me, they are a game changer in molecular biology. So before we begin with the lecture, let's have a look on the syllabus of the 13th unit and do the syllabus analysis. So in this unit, we are going to study about different aspects of recombinant DNA technology, starting from the isolation of DNA, RNA, protein, then the separation of these biomolecules using 1D, 2D gel, and also isoelectric focusing. Then the most important part is molecular cloning, in which we will get to know about different types of vectors used, different restriction enzymes, and like how the cloning is done. Then expression analysis, and then the genomic libraries construction, like genomic library construction, and also cDNA library constructions. Then the protein and DNA sequencing. Some other topics are also there, like in vitro mutagenesis and some of the molecular markers like RAPD, RFLP, and all of that. So let's begin with the basics. What is recombinant DNA technology? It's essentially a way to combine DNA from different sources to create something new. So imagine cutting out a specific gene from one organism and passing it into another. This method helps us produce important things like insulin or genetically modified crops. So now to break this down, some key tools are used in this process like restriction enzymes, which we are focusing on today, then DNA ligases, vectors, and the host organisms. Enzymes play a very important role in recombinant DNA technology, and there are different types of enzymes like restriction endonucleases, type 1, type 2, type 3, DNA ligases, reverse transcriptase, and some other enzymes like nucleases are used in recombinant DNA technology. All right, so let's dive into restriction endonucleases. So these enzymes cut DNA at specific sequences, which is why we often call them molecular scissors. They are found in bacteria and act as a defense system against viruses. So how? Well, when a virus invades, the bacteria use these enzymes to chop up the viral DNA and to protect themselves. Okay, so there are different types of restriction endonucleases, and the main types are type 1, type 2, and type 3. Each has its own unique properties. So now let's see that type 1 enzyme cut far from the recognition site and are not widely used in recombinant DNA technology. Type 2 is most widely used, and it is because of the reason because it specifically cuts at exactly at the recognition site, making them ideal for genetic manipulations. Type 3 enzymes cut a short distance from the recognition site and are used in some specialized applications, while type 4 enzymes target modified DNA and it has a role in epigenetic studies. So now type 1 restriction enzymes, these are a bit complex. Type 1 enzymes have both restriction and methylation functions. They cut DNA far away from the recognition site, sometimes more than 1,000 base pairs away. They need ATP for energy and magnesium ions to function. However, because they cut so randomly, they are not really widely used in the lab. Now, type 2 enzymes are the ones that are often used in molecular biology. Type 2 enzymes cut DNA right at the recognition sites, and their recognition sequences are usually 4 to 6 base pairs long. And what's even better is they don't need ATP. Just magnesium is required, and these are the ones which you use for cloning or DNA analysis. Type 3 enzymes cut about 20 to 30 pairs downstream of the recognition site, and they also need ATP and magnesium to work, but they are less efficient and rarely give complete cuts, so we don't use them as often. So now you know each and everything about it. And what you can do is just write the examples of type 1, type 2, type 3 restriction endonucleases in your notebook, and where they exactly cut, especially for type 2 restriction endonucleases. Here I have given one example of EcoR1. So it cuts at GAATTC, GATT-C. I often say that to memorize. So it cuts between G and A, and this is about the EcoR1. Similarly, you can check for other exams as well and write it down, because it may be asked in any exams, like it may be DBT or GATE Biotechnology or CSIR or your semester exams. It will help you. Now we are going to solve a question based on frequency of these sites in a genome. So before that, let's talk about how these enzymes know where to cut. They look for specific DNA sequences called recognition sites. For example, as I have shown you earlier, that EcoR1, one of the most commonly used enzyme in recombinant DNA technology, recognizes the sequence GAATTC and cuts between G and A. And what's interesting is that these recognition sites are usually palindromic, so means that the sequence reads the same forward and backward. Now, here is where things get interesting. How often do these restriction sites appear in DNA? So this is known as the recognition frequency, and it depends on the length of the sequence. So, for instance, if a restriction enzyme recognizes a 6 base pair sequence, the probability of finding it is 1 in 4 to the power 6. If it is of 8 base pair, then 4 to the power 8, and similarly. So you can see this question. So in a genome of 3 billion base pairs, a restriction enzyme recognizes an 8 base pair palindromic sequence. What you have to calculate is the expected number of times this sequence will appear in the genome. So that you have to do with 4 raised to n, and you have to and here it is 8. So if the enzyme cuts the DNA into fragments, what will be the average fragment length? And how would the frequency change if the sequence recognized by the enzyme was 7 base pair instead of 8? So I have given you the hint for this question. Now you have to solve this and write the answer in the comment section, and I will also post the answer in a few days based on your response. And if you like our content, I invite you to join our channel and subscribe it. So as the name suggests, bio exam and research support. So here we are providing support for bio exams like CSIR NET JRF, DBT JRF and GATE BT, and also the biotechnology semester exams for BSc, BTech, MSc, MTech students of biotechnology. So it will be very helpful for all of them. And apart from that, we are providing support for research paper writing, all the tips and tricks to write a best version of your research and review paper, and some PhD tips and motivation to keep you motivated during your academic journey. And one of the most important part, which is my favorite, and that's the study productivity. That on that particular topic I have made almost 27 or more than that video, so please go and check it. Thank you so much for watching till then. See you in the next video. Bye bye.